Isolating, screening, and identifying bacteria and fungi found within mangrove soil in South Florida for accessible antimicrobial natural products. Each mangrove ecosystem around the world has a unique microbiome within its soil. Many studies on different mangrove soils in countries around the world have identified organisms that produce antimicrobial natural products, but no research has ever been conducted on South Florida's mangrove soil microbiome. Secondary metabolites are organic compounds produced by organisms that aren't directly involved in growth, development, or reproduction, but often play crucial roles in survival. These molecules synthesized within cells include a vast array of compounds like alkaloids, terpenoids, and phenolics. Natural products refer to these secondary metabolites when they're harnessed for medicinal, agricultural, industrial, or commercial purposes due to their diverse biological activities such as antimicrobial, antioxidant, or pharmaceutical properties, making them a valuable resource for drug discovery, agriculture, and various industries. In the diagram shown here, you can see how the release of secondary metabolites from organism A caused the lysis of organism B. My research question is, can the microorganisms isolated from South Florida mangrove soil be used as a source of new natural products? My first hypothesis is that if bacterial and fungal species found in mangrove microbiomes around the world have been used as sources of antimicrobial natural products, then the bacterial and fungal isolates from South Florida mangrove soil can be used to develop new antimicrobial natural products. My second hypothesis is that if bacterial and fungal species found in mangrove microbiomes around the world are each uniquely diverse, then the South Florida mangrove soil microbiome has the potential to be as well. So first, in order to isolate the organisms that I obtained from the Florida mangrove soil samples, I had to follow the protocol which is shown here. I gave each organism an ID based on the agar that it was grown on and the order in which it was named, out of all organisms found on that particular agar. Once I had the organisms isolated, I then proceeded with a secondary screening procedure to identify the organisms that had antimicrobial characteristics. I performed an antagonism/dual culture assay, and the procedure is shown on the screen as well. After completing my antagonism/dual culture assay, I found that isolates B6 and E13 presented antifungal properties. Here you can see the zones of inhibition that each isolate created outlined in red. The zone of inhibition is where the model organism Aspergillus niger wasn't able to grow due to the excretions of each isolate. I decided to extract the secondary metabolites from isolate B6 and test to see its antifungal efficacy. Here you can see that the metabolites extracted from the isolate did show antifungal potential, as the average inhibition zone radius didn't significantly differ from the positive control amphotericin B. This was justified by a two-tailed t-test that generated a p-value of 1.78, which is greater than 0.05, showing no significant difference between the two groups. The DNA from the isolates was extracted and primed with primers specific to taxonomic kingdom. These tables show the identification of each sample. The samples were identified by metagenomic sequencing the extracted and primed DNA from each isolate. The species displayed in the graphs are based on the species within the samples that had the highest read percentages. The table shown on the left shows that of the 13 inferred fungal isolates, all 13 were identified to species level with 7 unique species. The table on the right shows that of the 10 inferred bacterial isolates, all 10 were identified up to genus level, 6 were classified up to species level, and 3 unique species were found. The unclassified species level of samples B3, B4, A2, and B6 can possibly be that the 4 samples were composed of undiscovered bacterial species. Here is a dendrogram showing the evolutionary distances between each identified organism up to species level within the Florida mangrove soil samples based on the alignment of the nucleotide sequences of each organism. The graph is measured by substitution per site, which represents the number of changes, mutations, and/or substitutions per site along each organism sequence. The yellow shaded portion is composed of the identified fungal species, and the purple shaded portion is composed of the identified bacterial species. The graph was analyzed using bootstrap analysis. Bootstrap values for all branch lengths were above 70, which shows their significance and accuracy in showing phylogenetic distance. It can be seen that the similar organisms clustered closely in the dendrogram, whereas organisms that differed clustered further from each other. Here is another dendrogram with the same setup as the previous one. The graph was analyzed with bootstrap analysis, and the branch values generated were all above 70 as well. This graph is comparing the evolutionary distances between organisms within the Florida mangrove soil microbiome and the Indian mangrove soil microbiome. Floridian species are highlighted green, and Indian species are highlighted red. It can be seen that the clusters of the Floridian species and Indian species remain separated. Also, the branch between the main Indian fungal cluster and the main Floridian fungal cluster is 0.08 units long, which signifies a large diversity sum between the two clusters. The bacterial clusters are also separated between the Indian mangrove soil species and the Floridian mangrove soil species. The results of this comparison can support the hypothesis that Florida's mangrove soil microbiome is vastly unique in comparison to other mangrove soil microbiomes around the world. In conclusion, 23 organisms within South Florida mangrove soil have been identified, with 7 unique fungal species, 3 unique bacterial species, and 4 unclassified bacterial species. Florida's mangrove soil microbiome showed novelty when compared to the Indian mangrove soil microbiome. Also, it has been confirmed that antimicrobial natural products can be found in South Florida mangrove soil. In the future, I would like to refine the antifungal products of the isolated organisms to create usable antifungals, isolate more organisms with potential antimicrobial characteristics, and explore further uses of the organisms identified. Thank you.